My name is Rushingisha George, working with Tanzania Fisheries Research Institute in Tanzania. The title of my talk is Seagrasses in Warming and Acidifying Oceans. Specifically, I'll be focusing on physiological responses. Seagrasses are marine flowering plants that complete their life cycle while submerged underwater. They are primary producers and ecosystem engineers in the marine environment, which substantially support ecological and ecosystem functions. Seagrasses are nursery grounds for most of fish species. Currently, however, seagrasses are being threatened by climate change, specifically increasing sea surface temperature. But climate change is also responsible for ocean acidification, which can be indicated by decreasing pH. Seagrasses are globally distributed with high concentrations occurring in the Indo-Pacific region, as you can see from the map. And by being globally distributed, seagrass vulnerability to warming waters vary with regions, and it is being said that seagrasses that occur in the tropical region are at high risk to ocean warming compared to those occurring in the temperate region, and this is due to prevalent climatic conditions in the tropical regions, which is high air temperatures and strong insolations. It should also be noted that intertidal seagrasses in the Western Indian Ocean, for example, are subject to tidal variability during the day-night cycle, where you find that high air temperatures and strong insolation heat the waters in the pools to 40 and 45 or even above degrees centigrade. Such high water temperatures can affect seagrass productivity, but also this can have impact on ecosystem functions and services. A small change in water temperature, for example, can affect substantially key physiological processes, for example, photosynthesis and respiration, which are determinants of plant survival, biomass allocation in a plant, the carbon capture and storage capacity within a seagrass meadow. Changes in key physiological processes can also trigger changes in biogeochemical processes, and under elevated conditions, for example, or disturbance, can lead to increased sulfide concentrations in the sediments, but also increased emissions of greenhouse gases such as methane and carbon dioxide. While sulfide is toxic to plant roots, methane and carbon dioxide, these are greenhouse gases which can add to the impact of climate change. Changes in plant productivity and increased biochemical processes in associated sediments can have significant impact on sedimentary carbon, which is the carbon that is stored in the sediments, in the seagrass sediments for long time periods, but also on the ecosystem functions and segments. Increase in temperatures above ambient levels can increase mitochondrial respiration and at the same time reduce photosynthetic performance of a plant, which could make a plant to experience high carbon demand with low carbon supply. This can lead to negative carbon budget in a plant, inadequate carbon in a plant can trigger the loss of stored carbon, which is also called carbon reserves, to balance the deficit of carbon. The loss of carbon reserves can trigger biomass loss in a plant, and the biomass loss can increase the loss of seagrass cover. When seagrasses disappear, they can lead to, or they can enhance losses of sedimentary carbon and ecosystem services such as nursery functions. Therefore, understanding of seagrass responses to future warming and acidifying oceans is very important. We have been conducting some microcosm experiments in our region to try to understand seagrass community responses to elevated midday temperatures, which frequently occur in the region, especially during midday hours, and they are expected to increase under future climatic conditions or under future climate. We tested, for example, 4 species of tropical seagrass plants to different elevated temperatures. We had 45 degrees centigrade, 40 degrees centigrade, and 36 degrees centigrade, 34 degrees centigrade, and the ambient temperatures, which ranges between 29 and 33 degrees centigrade. We were interested to understand the photosynthetic performance following temperature disturbances, but also how these how changes in photosynthetic performance affect biomass production. What we found is that seagrass photosynthetic performance decreases with temperature stress, and this was significant already from the first day of the experiment. As you can see here, for all species, there was a reduction in photosynthetic performance, especially under 45 degrees centigrade temperature treatment. Another interesting thing is that the effect of high mid temperatures, such as 45 degrees centigrade, frequently occurring in our seagrass meadows during spring low tides, increased with days of repeated exposure. And you can see here some species, for instance, Thrasodendron ciliatum, which mostly occur in deeper waters, survived only for 4 days, which could indicate that seagrasses that occur in shallow waters are more tolerant to high midday temperatures compared to those which occur in deeper waters like Thrasodendron ciliatum. And reduction of photosynthetic performance led to increased seagrass biomass, and you can see here from the results that high biomass losses occurred at high temperatures, 40 and 45 degrees centigrade, but still there were high losses of biomass at 36 degrees centigrade. And we also wanted to understand how biomass losses and the reduced photosynthetic performance at high temperatures affected biogeochemical processes in the seagrass sediments, and it was clear that methane emission, for example, increased with temperature stress. And interestingly, the effect of disturbance on emission of greenhouse gases lasted even beyond stress hours. As you can see, during night for temperature stress or at high temperatures, there was high emissions. This means that when we disturb seagrass meadows, their effects can also be seen during night hours when there is no stress, for example. And disturbances also led to increased production of sulfide in the seagrass sediments, where sulfide concentration was highest in the highest temperature treatment. And at the same time, the effect of disturbance was also seen during night hours when there was no temperature stress. And also the effect of disturbance seemed to increase with repeated days of exposure. To summarize, in the context of the WEO region, high midday temperature stress frequently occurring in seagrass meadows lead to decreased carbonification in the seagrass plants, which also trigger biomass loss or the release of carbon reserves to complement the carbon deficit in a plant. So reduced carbon fixation and increased biomass loss lead to increased methane emission and other greenhouse gases, but also they lead to increased sulfide concentration in the sediment, which on the other hand can also lead to increased biomass loss. The loss of seagrass biomass can lead to loss of seagrass cover and loss of sedimentary carbon, with huge impacts on ecosystem function. We should also be aware that seagrass meadows in the WIO region also experience other anthropogenic stresses, such as from coastal developments, dredging, pollution and eutrophication, seaweed farming, sedimentation, overfishing and beach seiling, which implies that these other anthropogenic disturbances increase the risk of WIO seagrasses to climate change impact. When it comes to seagrass response to elevated carbon dioxide and the climate change situation, there is a lot of debate which is underway now. Some scientists argue that seagrasses will be winners, and this is because elevated carbon dioxide can lead to increased seagrass productivity, but others argue that increases in carbon dioxide will have no effect on seagrass productivity. And taking that into mind, I would like to share with you on this ecosystem function of seagrasses, which have a strong influence on pH of coastal habitats. You should bear in your mind that seagrass meadows in the WIO region are subject to tidal variability, where they experience 2 high tides and 2 low tides during day-night cycle, and this can have effect on functions of seagrasses, especially looking at the pH of other adjacent ecosystems. We measured pH in coastal habitats of Chwaka Bay and Mnazi Bay. These are spatially isolated bays in Tanzania, which harbors coral reefs, mangroves, and seagrass meadows. And the mean pH between habitats showed that highest mean pH were recorded in seagrass meadows, and this was followed by coral reefs. And looking at the pH of coastal habitats during high tides and low tides during daytime, it was clear that highest pH were recorded during low tide conditions in seagrass meadows, and such pH were buffered or reduced during incoming high tides, where you can see that for the case of seagrass meadows, pH was reduced when you compare to that of the days low tide. But interestingly, if you look on coral reefs data and the mangrove data, you can see that during the high tide, there is some sort of pH increase, and this informs that seagrasses can support or increase pH of adjacent ecosystem, which is very important when it comes to the impacts of ocean acidification. To conclude, I'm saying that intertidal seagrasses of the tropical weir region are at high risk of declining under future global warming conditions, and this is due to prevailing climatic conditions, which are high temperatures that can combine with strong insolation to negatively affect productivity of seagrasses, especially during midday hours. Also, disturbances on seagrasses will lead to increased emissions of greenhouse gases and can change seagrass meadows from being sinks to sources of atmospheric carbon with higher impacts on climate change. Seagrass meadows increase pH of adjacent coastal habitats and can help to mitigate the impacts of climate change, especially ocean acidification, on short time scales. I'm ending by saying reducing human stressors on coastal ecosystems can increase seagrass resilience to ocean warming while mitigating the impacts of ocean acidification. Thank you for listening.